Hello, everyone. We're back again from one more talk about the series of the molecular methods applied to animal ecology course with Dr. Drinkwater here. It's a big pleasure to have her here. She does a lot of exciting stuff, like a lot of exciting research. So Rosie is a postdoctoral researcher working at Queen Mary University of London and the Natural History Museum also in London. Overall, she's interested in how we can use DNA to understand biodiversity, conservation, and the impact of habitat loss in the tropics. She's currently working on the project related with conservation genetics of two endangered mammals in Sulawesi, Indonesia, the babirusa and anoa. I hope I have pronounced their names right. For this, she's using ancient DNA from museum specimens to improve spatial and temporal sampling of this taxa to ultimately understand the impacts of recent anthropogenic deforestation. For her PhD, she has used DNA from leeches to survey mammals in a human-modified landscape in Borneo, Malaysia. She sampled blood-feeding terrestrial leeches from longer gradients of forest degradation to understand the usefulness of this technique for rapid biodiversity surveys. She's involved in a lot of field work as well in forests in Sabah, Malaysia, Borneo, as well as a lot of time she spends also in the logged and degraded forest habitats. She has been adapting as well techniques for sampling mammals with DNA from the guts of dung beetles, which is tricky because DNA is even more degraded, having essentially been digested twice. Over the years, she has done a master's as well and worked as a technician with working with most of the samples of environmental DNA, but also she has worked with surveying invasive crayfish in water samples in the UK to categorize and also categorizing the samples, the diet samples of lemurs for fecal samples in Madagascar as well. So it's a big pleasure to have here Dr. Rosie Drinkwater talking about blood and guts, invertebrate derived DNA from leeches and dung beetles for biodiversity surveys.
Okay. Can you hear me? Is that okay, Hanani? Yep. Okay. So thanks for the introduction, Hanani. I am going to be talking about surveying mammals using invertebrate-derived DNA. And this is part of my PhD work that I did a few years ago at Queen Mary University in London. So first, I'm just going to start with acknowledgements because there's a lot of people that helped with this work. So my supervisors, Steve Roster and Beth Clare, and then all the co-authors for the studies and technicians and institutes which provided permits and funding for all the sequencing and fieldwork. And I'm not sure if any of you had done fieldwork before, but we all know how important research assistants are in the field, so they need to have a big thank you as well. So in this talk, I'm just going to cover what is iDNA, where do we get it from? I'll introduce the study site where I did most of, all of the field work, which is the state of Sabah in Malaysia Borneo. I then will quickly go over the PCR, the general methods for sequencing and PCR, and then how I used these techniques to answer specific research questions. So mainly comparing two different types of leech species and what they feed on, conducting a biodiversity survey across the field site in Sabah, and then some preliminary work I've been doing recently, which is regarding getting iDNA from new sources. So we've been using dung beetles. Okay, so what is iDNA? So I call it invertebrate-derived DNA, but I've also heard ingested DNA. And it's you then amplify and sequence target species DNA from the gut. You sort the sequences bioinformatically, and then you use this to identify the taxa. And you do all this without having to see your target species of interest. And that's why exactly why we use iDNA, because that's the benefit, that you can remotely identify species without having to see or hear them in the forest. And this is really good in the tropics because you have many rare species or elusive species, but they also have a whole community of invertebrates that feed on them, on their blood or on their dung or on like carcasses or open wounds. And this all contains DNA of the host. Invertebrates are always, tend to be a lot easier to catch than mammals. They're more abundant. They tend to be generalist feeders, so that if they feed on mammals, they'll probably feed on all the mammals. And from the point of view of the mammal or the large vertebrate, it's non-invasive. Obviously, it's very invasive for the invertebrate. And all this gives us access to rare species which we might not normally be able to see. It requires less taxonomic expertise in the field. And also because of the speed of field sampling, we can cover a really wide area in a shorter amount of time. And it's really great because at the moment people are combining it with other field sampling techniques, so with camera trapping, and also combining it with analysis pipelines like occupancy modeling. So these are the two samplers of choice. I'm just going to stick to the common names at the moment. So this is the brown leech and the tiger leech, and they're both hemodipsid leeches. So this is, they are terrestrial blood feeding leeches, and it's a whole family, the Hemodipsidae. So this was the first paper by Ida Schnell in Copenhagen in Tom Gilbert's lab, who first pioneered this, this way of sampling mammal biodiversity using a similar hemodipsid leeches, but from Vietnam. And their first paper, basically, they identified the window of DNA detection. So they fed leeches in the lab, I think it was goat blood, and then using qPCR, they could track the DNA decay with time post-feeding. And as you can see, you can detect DNA in the leech's blood like a really long time after feeding. So we're 60 days, 80 days. And this means that if you go out and randomly sample wild leeches, you are more likely to be able to detect their last blood meal, which is great because you don't know when they last fed. And this is exactly what they found. So they went out and took a small amount of leeches from these field sites in Vietnam. And 21 out of the 25 samples had a positive hit for a mammal. And they were looking for the very rare antelope, the saola, which they didn't find, but they did find other pretty rare species, including the Annamite striped rabbit and the ferret badger. So yeah, basically this study demonstrated how the potential of leeches for identifying mammals in this way. However, leeches, well, the hemodipsid leeches, which are the blood feeding ones, they only, they have quite a restricted range. So they live in South and Southeast Asia and Madagascar and Australia. And that's kind of it. So if you want to do your sampling in a different place, Brazil or South America, then there's other invertebrate samplers that you can use. So there's lots of studies that have come out recently which have used carrion flies or blowflies, which feed on like open wounds or carcasses or feces, which you can use the same pipeline to detect the mammals that they've been feeding on. And this is true of mosquitoes and also of sandflies. So basically anything that's been feeding on the vertebrates. So I'm just going to introduce you to the field site. So this is Southeast Asia. And here we have Borneo in the middle. So it's governed by 3 countries, Indonesia in the south, Malaysia in the north, and Brunei is just nestled in the middle in the north. All my field sampling was done in Sabah, which is in the northeast state, where I've circled. And this whole region is being, lots of the forest is being converted to oil palm from primary rainforest. And because of this, there's a need to understand how these degraded landscapes harbour biodiversity, because primary rainforest now exists, especially in Borneo, tend to only exist in protected areas already, but there's lots of sort of logged or degraded habitats which risk being converted to plantation and may hold a higher conservation value for biodiversity in their degraded but forested state. So these are my 2 study sites. The Danang Valley Conservation Area, which is a primary old growth rainforest. It has extremely high levels of biodiversity and very low levels of human impact and low levels of hunting. And then in contrast, there's the SAFE project, so the SAFE project stands for Stability of Altered Forest Ecosystems Project. And this is a large-scale fragmentation experiment. So we'll just zoom in a little bit into the SAFE project. So I'm not sure if you can see, no. So this is a whole region that's being converted into oil palm plantation. And while the conversion is ongoing, there are these long-term vegetation plots, which you can see as the white dots on this map. And these are fixed vegetation plots where people are monitoring biodiversity, ecological processes as the landscape is being converted. And so at the top of the map, you actually have like the control forest, and this is twice logged forest. So it's been selectively logged. Most of the large trees have been taken already. But it's still quite good habitat. And then in the middle, you have the heavily logged and pretty degraded forest habitat. And then surrounding all this landscape is oil palm plantations. And this mosaic landscape is very typical of Borneo and Southeast Asia. So these are just some pictures just to show you what the forest looks like. This is Danang Valley. This is the old growth rainforest. And as you can see, it's all tall trees, the canopy's continuous, which is compared to this. This is the SAFE project. So there's lots of evidence of human infrastructure for logging and for oil palm. There's roads, there's logging landing sites where they store all the logs. And basically it's pretty trashed forest. And then these are the oil palm plantations, which this is a downside of sampling with leeches, because we know that especially some of the big mammals use oil palm plantations to connect fragments in the landscape. However, it's so hot and dry inside oil palm plantations that you cannot, it's too inhospitable for the hemodipsid leeches. So sadly, we didn't actually sample there. So this is just a general, how do you sample for leeches? So at these 25 meter squared vegetation plots, we did hand searching within the boundaries of the quadrat and we searched for 20 minutes. And leeches are quite good because all you need to do is rummage around and they tend to come to you. And then you can collect them, put them in RNA later, which is for stabilizing DNA and RNA. And then we aim to keep them cool, which because the rainforest is so hot, and this involved just carrying around a cool box everywhere I went to keep the little tubes in. And we wore gloves at all times to try and minimize the amount of human DNA that we were putting on the leeches. And also we never took a leech if it had already bitten and fed on a human, because there's absolutely no point, because you're just going to get human DNA. So just an overview of the methods. You probably know quite a bit of this already. But what we did was we individually extracted each of the leeches and then pulled them together. And we did this so that we could maximize the amount of leeches we could get sequenced. But then also we could go back to the individual leeches, say we needed to, or we found something interesting. That was always a possibility. We used PCR, so we were targeting a very small fragment of the 16S gene, and we were using primers that had been generated for ancient mammals, which is why they were really good at picking up highly fragmented and degraded DNA. Then our pools of leeches was, the amplicons were sequenced using Illumina sequencing. This sequence data was then filtered for quality and length. And then we used an OTU clustering method, and there's lots of different ways to do this. And at the time we were using OTU clustering. So OTU stands for Operational Taxonomic Unit. And this just means that the sequences were clustered based on sequence similarity. I think most of the time I used a 95% similarity filter. We also then added an OTU filtering step, which just cleaned out any OTUs that had arisen from sequencing error. And this is because we really wanted to be conservative about we didn't want to inflate our biodiversity estimates because of sequencing error. And then we used BLAST to identify the mammals, and we were quite lucky in Borneo because the mammal species are quite well defined, apart from maybe the small mammals, but definitely the big mammals. There's lots of sequence data out there. We generated a bit for our first paper as well to add to the database. But so we built our own bespoke reference database, which only contained Bornean mammals plus any lab contaminants that we thought we'd need to pick up as well. So using these methods, I was answering 3 main questions. So the first one was looking at, so I have the brown leech and the tiger leech, and we're looking at the feeding differences between these 2 species, and also just the occurrence across the landscape. So is it different? Do you find them in the same places? So here we are again, brown leech and the tiger leech. And very little is known about these leeches. Very little work has been done on them. When you're in the field, you realize quite quickly that they've got quite different behaviors. So the brown leech lives in the leaf litter and is very you don't see it very often. And it'll have bit you and your foot will be bleeding before you know that you've been bitten. Whereas the tiger leech is really obvious and hangs on leaves and in the understory between the ground and 2 meters, and it's quite aggressive and it will really come for you. So based on these behavioral differences, you could imagine that maybe they were feeding on a different suite of the mammal community. And it's quite important to understand your invertebrate sampler behavior because you want to know if you're skewing the biodiversity estimates one way or another. Yeah, okay. So using that hand searching technique at the vegetation plots, we had 2 habitat types here. So the heavily logged forest, the really poor quality forest, compared to the twice logged forest, which was the higher quality forest. And we were collecting brown and tiger leeches at both sites, or at sites within each habitat type. And like I said, we individually extract all the leeches and then pool them together. So I think these are all pools of 10, yeah. So first, if we just look at species accumulation, so this is the accumulation of the mammal species that we're identifying in the leech diets. You can see quite obviously that the tiger leech is always detecting more mammals for the same number of leech pools sequenced as the brown leech. And then just looking at the occurrence of the mammals in the diet. So here, these are quite common species. You've got the bearded pig, you've got the deers, so the samba deer, the muntjac, mouse deer, and then the moon rat, and they're all being detected in roughly, not the same, but high proportions in both of the leech species. And then you have this group down here where you're seeing more detections in the tiger leech. So this is the primates, the gibbon and the macaques, and the civets, and also the rodents, including all the porcupine species. So we are seeing a difference really between the diversity and accumulation of mammal species, and it seems to be that tiger leeches are feeding on more mammals and a different suite of mammals. So then we, based on this need to understand the ecology of the invertebrates, we built site occupancy models. So these can get quite complicated, but basically we were comparing the occurrence data of each species of leech from 2 different seasons, because I sampled in the wet season and the dry season. And occupancy models are based on detection and non-detection data from repeated surveys at the same site. And there's 2 things that you just need to take away from this, is that occupancy probability is the probability a sampling site is occupied during the season, and detection probability is the probability that you detect the species given that it occurs at the site. Okay. So we built lots of models, and we basically were looking about how occupancy probability and detection probability vary with certain measures of habitat covariates. So let's look at these. So in this slide, the brown leeches are at the top always, and they're in grey, and the tiger leeches are at the bottom, and they're in yellow. And so these are showing the distribution of occupancy probabilities dependent on the habitat covariates. So you have canopy height, and which is just literally how high is the canopy, and habitat heterogeneity, which is a measure of clustering. So the higher the value, the more fragmented the landscape is, the more clustered together. And canopy height was always retained in all the models, but there was quite a small effect size. But we found that in the dry season, the brown leeches showed this quite a strong negative effect with habitat heterogeneity. So occupancy probability decreased for brown leeches the more clustered and fragmented the habitat got. We also then looked at detection probability. So this is the probability that you detect the leech given that it is at that site. And I'll just pull out some of the important ones. We see that for both species in both seasons, that canopy height is the biggest factor on whether we're going to detect the leech there or not. So you're more likely to detect the leeches when the canopy is high, basically, which is likely to be an interaction with microclimate, because these leeches like it, hot and humid, and open canopies make it drier. I'm going the wrong way. We also see here, like the brown leeches before, but this is an interaction between detection probability and clustering habitat heterogeneity in the wet season for the tiger leeches. So you are less likely to detect them in more clustered habitat during the wet season. And what we're seeing there is that these two leeches are not just behaving exactly the same species. They're not behaving as exactly the same. And so it's really important. I think that really drives home the point that we need to understand the ecology of the invertebrate is going to affect your biodiversity estimates. Yeah. So, and for this reason, when we conducted our large biodiversity survey across the habitat modification gradient, so at the different habitat types, we stuck to just using tiger leeches because they were detecting a wider community of mammals. So this was the second study where we were looking in the twice logged forest. So this is the higher quality forest. It's been selectively logged, so there's no big trees there really anymore, but the canopy is quite closed. There are signs of damage, there's lots of lianas, but generally for the area, it's quite good habitat. And this is compared to the heavily logged forest, which is like those photos I showed you before, where it's really variable habitat. So you have the really fresh, freshly logged sites where the trucks and the tractors have just been taking logs out. And then if a site has been logged quite a long time ago, you start to get this regrowth and it starts to look quite green. It's just there's no trees and no canopy, basically. And again, we compared this to the old growth forest at Dannon Valley with these massive trees, and Dannon Valley actually currently still has the highest, tallest tropical tree measured to date. There is one caveat with this, is that we only had, whereas we had multiple sites within the other two habitats, we only had one old growth habitat at Dannon Valley. So again, this is just a breakdown of the pools and the individual leeches that we collected. So this is all just tiger leeches, and you have the 3 habitat types. And we can see just off the bat that you are getting slightly more mammal detections in the twice logged forest than in the heavily logged forest. The old growth is maybe a bit misleading because it is pretty undersampled. So this is just, again, the number of detections for each habitat type. And we can see that rusa, which is the large sambar deer, and sus, which is the bearded pig, they're detected in, and the muntjac, they're detected in all 3 habitat types. So purple is the heavily logged site, light green is the twice logged site, and dark green is the old growth site. We also see mouse deer, that's only detected in the twice logged site. And we see quite a number of hystrix detections, which is a genera of porcupine. However, we were unable to distinguish between the 2 species of hystrix at this 16S site. So that's another sort of limitation of the study. We also surprisingly detected some pretty rare species. So we found a pangolin, which is the manus, the genus manus, which was detected in the twice logged forest, and the sun bear. Again, with the sort of discrimination of the 16S marker, we found a hit to a cat, or the cat family, the Felidae, but there's multiple cat species in Borneo, and there was very little confidence around this detection. But interestingly, it was in the heavily logged forest. And we also get a really wide range. So the bottom 4 genera are all civets, and there's 5 genera of civets in Borneo. So we got all but 1. So what we did with this study is we combined these diversity detections with hill numbers, which basically are equivalent to species richness, Shannon Diversity Index, and Simpson Diversity Index. And what we can see is in the solid graph, where the bars are solid, this is like an estimator, a diversity estimator. And the twice logged forest site has a higher diversity than the heavily logged forest site. However, as I alluded to earlier, there's probably quite a difference in sampling effort. So, and once we rarefied this diversity, so we rarefied the sampling number, we see a more uniform detection, even with species richness. And this is the same with the next order of Hill numbers, so where Q equals 1, which is equivalent to the Shannon Diversity Index. We see even in the non-rarefied data that this starts to become more similar. And it shows that the difference between the twice logged and the heavily logged sites is probably being driven by rare species or unevenness, basically. And this generally is quite good because it shows that we're still getting high levels of diversity, or high enough levels of diversity detected in these really heavily degraded sites compared to the twice logged higher quality sites. So these are NMDS plots. So this is a non-metric multidimensional scaling plot. And these are based on community dissimilarity at the different sites. So the green circle is the twice logged forest community, and the purple again is the heavily logged. And there's only one site for the old growth, which is the one labeled OG. So you can't really do much with this. But what you can see from here is that if you look at just the first and second axes, the heavily logged forest community is nested within the twice logged forest site. But when you start looking at the second and third axes, they're showing some differentiation between the two communities. So basically, this study showed us that we're still getting diversity in the heavily logged forest sites, and they potentially have a slightly different community composition, which is all great because it shows you that the conservation value of these heavily logged sites is still quite high. And finally, this is just some preliminary research that we've been doing. Basically, where we've been attempting to extract iDNA from dung beetle guts using the same metabar coding pipeline that we had set up for the leeches. So there are some benefits of using dung beetles over leeches for iDNA. You can trap for them in a standardized way. So as opposed to waiting for the leeches to come to you, the beetles are being drawn towards a trap, which you set with a dung ball, which they're attracted to. They're less habitat restricted, so you find them in the oil palm. So where we don't have that extra comparison, like what happens when you convert that heavily logged site finally into an oil palm plantation, you can't see that with leeches, you can't do that comparison. But with dung beetles, you would be able to do this. They also have a wider global geographic range. So where, like I said earlier, the hemodipsid leeches are restricted to sort of South and Southeast Asia, Australia, Madagascar. Dung beetles are found on all continents apart from Antarctica, so they're found everywhere. However, we are expecting more highly degraded DNA, so even shorter fragments of DNA, because it's essentially been digested twice. So this paper was just, we just conducted 2 experiments. So the first one I've called a controlled feeding experiment. So this is very similar to what Ida did in the first leech paper, where we wanted to see how long after feeding we could still detect DNA in the guts of the beetles. So we did this in the field where we live trapped Cataseus, Cataseus renopouliani, which is the largest dung beetle species in Borneo. Oh, well, both of the Cataseus species are. And And we did this basically so that we could extract the guts, dissect the guts out of the larger species. Some of the dung beetles get very, very small, so individually extracting guts starts to get a bit messy. We kept them in a box and we fed them cow dung for as long as they needed. And we then stopped them all feeding at the same time point and then dissected a set amount of their guts at 10 different time points after feeding. And then we analysed the DNA copy number using qPCR, which helped us to identify this window of DNA detection. So these plots just show, so A shows the log DNA copy number with the time post-feeding. So there's 10 time points where we dissected the guts of the dung beetles. And we can see a strong decay with time, but if you remember the original leech graph was like 60 days, we're talking hours here. So it's a much shorter window, and this is reflected in their feeding behavior. So leeches feed once, drop off, and probably don't feed again for quite a long time. Whereas dung beetles are always actively searching for their next meal. We analysed it, but we didn't find any relationship between sex of the beetle. So there's no male or female specific response, or gut weight, which we kind of expected gut weight to have a role, because you could imagine that a beetle that's just fed would have a heavier gut. But it's good in a way that we don't find this relationship, because it means that if you're sort of going to do these surveys, you don't have to bother either identifying the dung beetles to sex or weighing guts. You can just take anything. So the second experiment we did was we collected some wild beetles, again from live traps, and we either dissected the guts of the large Cataseus beetles, which we dissected in this field lab. So this is in Borneo, and it's basically just a plastic box sitting on some plastic cups. But it was to try and minimize the amount of human DNA or any other DNA that we were getting in there. And again, this was sort of to test whether it was possible just to do everything in the field, because if these assessments are going to become useful, we need to be able to do everything, maybe without sending samples everywhere on dry ice. We also took a few traps of the smaller beetles and just mashed them up just to see if we could get anything out of that as well. And it was just a very small number of samples, which we sequenced using the 16S amplicon pipeline, same as the leeches that I showed you earlier. And so this is, again, just a very preliminary experiment, but as you can see, we're detecting the same common species with a high read count. So this is after all the filtering steps as well. So you're getting bearded pig, samba deer, which are really, they're the most common species in the area, the muntjac, mouse deer, and the porcupine as well. We also got the banded civet, which you can see has a much lower read count. So we would be cautious about whether this is a true detection. However, you could imagine that it's just maybe a beetle that fed a much longer time ago on the civet dung. However, yeah, we would be cautious about this detection. But it at least demonstrates that it's possible to extract iDNA from dung beetles in the field. So in conclusion, basically, I think I've shown that iDNA studies with leeches and dung beetles, maybe there's probably a bit more work to do, have the potential for doing these rapid biodiversity assessments. And I say rapid just because you could do a week in the field collecting leeches compared to leaving camera traps out for months. So, there's lab work to do, which makes, it's different trade-offs really. But with all these things, there's really the need to understand how the invertebrate ecology is affecting your biodiversity estimates. but we are able to detect these common species, these large common mammals, as well as the rare species like the pangolins. It would be great, and there's lots of studies now where people have been validating these iDNA results with camera trapping data, which is great to combine the two. And like I said earlier, it would be great if we could do this with multiple markers, which would help us discriminate between some of those taxa where we couldn't say which species it was. So like the muntjac, there's 2 species of closely related muntjac in Borneo, and the hystrix porcupine. And as well, recently I've seen studies where they're combining invertebrate samplers. So they're doing flies, multiple families of flies, or carrion flies and mosquitoes. And then the aim would always be to go beyond just counting species in a forest and do something where we can combine it with occupancy models, or like combining it with the hill numbers, just to be able to answer questions about what's the value of these degraded landscapes or any other habitat that you're working in. So thank you for listening. I'll just leave you with this picture, which is basically the only leech that ever got me before I got it. But thank you for listening, and I'll take any questions.